So my lab uses the larval zebrafish to try and understand how information about the visual world is encoded at the level of retinal ganglion cells. So these are the output neurons of the retina. And we're also interested in understanding how information from the retina is distributed to the various visual targets in the brain. And in larval zebrafish, the main visual target is the optic tectum. And the tectum receives input not just from the retina but also from other sensory modalities, and it uses this information to drive movements of the eyes, the head and the body towards or away from visual targets. And so for a larval zebrafish, the tectum is crucial for generating behaviours such as prey capture and predator avoidance. And the retina is connected to the tectum via the axons of retinal ganglion cells, and these innervate the tectum in a number of very precise and stereotyped layers. And laminar organisation is not unique to the fish tectum. Layers are found throughout the brain, in many different areas of the brain, and also in many different species. And it's really not clear why the brain is organised in this way. So in a study now published in Neuron, a postdoc in my lab, Nikolaou, has used the zebrafish tectum to try and understand the significance of layers in the brain.
To investigate the role of lamination in the brain, I took advantage of the astray mutant zebrafish, which carry mutation in the axon guidance receptor robot 2. In the astray mutant, the precise laminar architecture of retinal ganglion cell inputs to the tectum is disrupted. We exploited this phenotype to examine how spatial disorder in the retinal ganglion cell inputs impacts the functional development of the tactile output. We focus our study on the development of retinal tactile connections that encode the information about the direction of motion, the so-called direction-selective circuits. To study retinal inputs to astray mutant tectum, I have generated mutant zebrafish that express a genetically encoded reporter of neural activity GCAM3, specifically in retinal ganglion cells. This has allowed us to functionally categorize retinal ganglion cells targeting the tectum and map the spatial distribution of functional subtypes within the tactile layers of wild-type and astray mutant zebrafish. We find that in both wild-type and mutant animals, there are 3 functional subtypes of direction-selective retinal ganglion cells innervating the tectum, which respond to motion in the upward, downward, and forward directions. By generating functional maps of these inputs, we show that in wild-type animals, axons of direction-selective ganglion cells innervate precise and stereotyped sublaminae within the superficial tactile neuropil, confirming previous findings from our lab. However, in astray mutants, the laminar organization of these responses is lost, and direction-selective responses are diffusely distributed in the tactile neuropil. These experiments show that the mutant tectum receives intact direction-selective signals from the retina, but these signals are being delivered to the wrong tactile location, that is, they are no longer laminar. How does this impact the functional development of postsynaptic tactile neurons? To address this, we generated a stray mutant with a pan-neuronal expression of GCAM and performed functional imaging of tactile neurons. The surprising finding was that in a 7-day-old tectum, direction-selective responses are normal. In both wild-type and mutant tecta, we find 3 subtypes of direction-selective tactile neurons whose preferred directions match those of the retinal ganglion cell inputs, and 1 emergent population tuned to posterior motion. However, this result does not mean that wiring specificity is maintained in the mutant. It could well be the case that in the mutant tectum, retinal ganglion cell axons are contacting tactile neurons that they would not normally contact, and these neurons simply inherit their responses from the misplaced axons. To test whether direction-selective retinal ganglion cells are still contacting their normal tactile neuron targets within the tectum, we targeted the expression of GCAM6 in single direction-selective tactile neurons using the FOXP enhanced array. This allowed us to examine both their tuning properties and morphology. In wild-type tecta, tactile cells tuned to forward motion have laminar dendrites that co-stratify with the axons of direction-selective retinal ganglion cells. When we label cells in the mutant tectum, we find that they retain their tuning to forward motion, but the laminated structure of the dendrites is lost. However, when we compare the distribution of direction-selective retinal ganglion cells and direction-selective tactile neuron dendrites in the mutant, we find a striking degree of overlap. Thus, it seems that structural plasticity within tactile circuits enables direction-selective retinal ganglion cell axons and dendrites of direction-selective tectum neurons to locate one another even when lamination cues are lost. We wanted to understand precisely why the morphology of tectal dendrites is altered in the astray mutants. It could simply be that because the presynaptic inputs provide the growth scaffold for dendrites. If retinal ganglion cell axons are disrupted, then so are the tectal dendrites. Alternatively, ROBO2 may play a role in directing the growth of dendrites. In addition to the expression in the retina, ROBO2 is also expressed in the tectum, but its role there is unknown. In our study, we found that ROBO2 does indeed guide lamination of tectal dendrites independently of retinal ganglion cell axons. Our results so far suggest that ROBO2 guides the growth of direction-selective retinal ganglion cell axons and direction-selective tectal dendrites to a common lamina. But when lamination cues are lost, as they are in the astray mutant, functional connection can still form between the appropriate cell types. An obvious question then is, why is lamination necessary? One idea is that by guiding the growth of retinal ganglion cell axons and the postsynaptic targets to a common lamina, ROBO2 facilitates contact between appropriately matched cell types. This could potentially speed up the process of circuit assembly. To test this idea, we performed functional imaging of tectal neurons much earlier in development. In wild-type animals, we find that even at these young stages, direction-selective responses are already well established. However, in astray mutants, at the same stage, there was a profound deficit in the development of direction-selective circuits. Thus, it appears that while a layered neural architecture is ultimately dispensable for the correct wiring of direction-selective circuits, lamination is essential for the rapid functional development of these neural networks. Our study provides novel insights into the development of retinal tectal connections. Our findings suggest that ROBO2 is an organizer of the tectal neuropil and acts to bring together retinal ganglion cell axons and dendrites of target neurons into alignment in precise and stereotyped layers within the tectal neuropil.
And I actually think one of Nikolas's most important findings concerns the role of layers themselves. We find that rather than being necessary for establishing wiring specificity, he finds that layers are absolutely necessary for the fast functional wiring of neural circuits. And if you're a larval zebrafish that's crucially dependent on vision for survival, then speed really matters.